Hi everyone, and very good morning, and welcome to the CellStrat Unplugged session. So we'll be having a second um, continuation for the part two, basically for the previous session that we have taken on AI-powered drug discovery and decoding disease with LLMs. So some of you may have attended the previous sessions, or some of you may not. So if you have any questions, please let me know. I'll just have a quick recap of what we have done in our previous session, and then we can go ahead on continuation of what we have done before. Okay, so this is basically we are trying to understand the LLM's scope in terms of drug discovery and disease identification process. So this is very much like an in-depth webinar that is consisting of multiple weekends, and I'm going to have this is like third week discussion, and in this we have like the primary topic today is like AI powered drug discovery, decoding disease with LLMs. So I'll be going through again the paper that we had discussed last week, and in continuation of the topics that were remaining, right? So just to quickly make you guys aware of what we have done last week, and that would set the context, and it will also warm up everyone to move ahead on this learning process or learning journey. So my first webinar in this series was about Gen AI and LLMs in the biomedical biomedicine in health, and then we have decoding disease with LLM. So this is part one is done last week, and this week we are continuing with the part two of the same paper. All right. So the paper name is, I had shared the link with you, all of you earlier, which is large language models in drug discovery and development from disease mechanism to clinical trials, right? So this is the paper, and if you guys want the link again, I'll just share it in the chat one more time, just to have that handy at your end, and if you wanted to refer it later, you can do that. All right, so this is the paper that I'm referring to. Okay, so in this we have I will like quickly cover what we have discussed in the last webinar, like uh, LLMs in drug discovery and development process. We had talked about two approaches, one that is using an specialized LLM, and the other one is that is using a generalized LLM. So specialized LLM versus generalized LLMs, and we cannot say that one replaces the other, because both has their own purpose. Some of the steps in drug discovery process needs and specialized LLMs. Like you can look at these here, that we have a molecule smile. So basically that understand the scientific language like biology or chemistry or protein or gene, and then the model is trained with these set of genes, like we have protein GPT or gene GPT, let's say, and that goes like an trained model, and then you are able to do certain tasks. So you basically train the model on a specific task, like retrosynthetic planning or reaction prediction, or protein ligand binding prediction, or de novo molecule generation, right? And there are some protein related tasks like protein structure prediction, de novo protein generation, and gene related tasks like gene network analysis, biomarker analysis. So today we will go in deep dive into these aspects, because in the last session we got an overview of an understanding of what are the specialized NLP tasks that are associated in the healthcare domain. And these are the usage of these, right? So once we are able to get this, a task specific model created, we are able to do, basically able to predict the protein ligand binding affinity score, or we are able to do further things along with that. And there are the other area, that is the generalized LLM that we already discussed about. Uh, the use of LLM is allowed by USFDA. That's a question by Ravi. Ravi, so these areas are like, already evolving, and if you see the Food and Drug Safety Board, they actually say that they have authorized 950 AI/ML medical devices and also the LLMs in that area. So it is basically evolving area, and still like a lot of approvals and things, regulations are going on, but I think over the time it will happen, if not now, immediately. Okay, so moving on, these these are the generalized LLMs that are trained with specific data set or a specific tasks that you can see, like more of like an generalized LLM, and not using any scientific language here, right? So it can be Wikipedia, or it can be clinical data, or it can be any textual references for the healthcare, from the healthcare domain or non-healthcare domain as well, right? So that's what is generalized LLM means. And we also talked about that it can be good for prior knowledge understanding, or you can train for reasoning or planning or tools or information retrieval, and you can build an assistant out of that. Okay, make sense? So these are the two aspects that we had discussed in our last webinar. Okay, cool. So if we move on, the next thing is that we are going to deep dive into the LLMs that are more of supporting this drug discovery and clinical trial process in the further understanding of it. So coming back to this, the main the paradigms that we talked about in our last session as well is specialized large language model like ESM1B, 1 billion transformer, or CameCrow, or GeneFormer, or LLM 4SD, or MedPalm, right? So these are some examples. There are so many LLMs that are already had been and are coming up in this space, and there are general purpose LLMs like GPT-4 or Galactica, right? So these are some examples of the general purpose LLMs. Okay, any questions, anyone, before I move on to the next set? Okay.
There was a question whether is FDA approved LLMs.
Yeah, I think I have answered that. Don't know if you
Sorry,
have.
yeah, or maybe I missed that. Sorry.
Yeah. When you say drug discovery, is it API? Drug discovery is not an API, Ravi. It's the process. It's not a tool yet, unless somebody builds a tool. Right? Drug discovery is the process of discovering the drug. Does that help? Oh, okay. Drug discovery, is it API? Not, I'm not sure, Ravi. These are like some of the clinical terms you are using. I am an AI, more of an AI expert than knowing the abbreviations, but I'm sorry. So you are talking about active pharmaceutical ingredient, is it? Active ingredient in the new drug. So this is more of a general drug discovery process, and not necessarily, I would say, specifically identifying only the active or APIs that are active drug discoveries, right? So, um, but we can, I mean, I think this is like more of an exploration area yet, because with the LLMs so far the drug discovery process is not an established solution. So I I kind of think that APIs would take a little bit of time, but I could not confirm that, Ravi, as of now.
I mean, GenAI, this drug discovery has hundreds of steps. So we are
Yeah.
going to discuss how GenAI can intercept some of those steps for value addition. So
Yep.
that's the goal. So the ingredients will remain what they are, but how to accelerate or optimize, drug discovery, instead of a drug discovery taking 10 years, can we do it in 6 months? That will be the goal of AI interventions in various hundreds of steps of drug discovery. So we'll be discussing that in these series of webinars, and we have a course lined up on AI interventions in drug discovery process.
Right.
Many courses lined up, in fact.
Yeah. So the idea here is to basically understand from the AI and LLM perspective what are the specific areas that we can cover in the healthcare domain. And one of the aspect that I'm covering in this webinar is going into the healthcare space, where we are talking about the drug discovery and development. Right. So these are the things that I'm going to discuss today, is about understanding the disease mechanism, right? Um, okay, I think Ravi is asking to explain again.
Well,
Vivek,
I'm not sure. I mean, there is not much to explain. So we are
yeah,
starting a series of webinars and courses where we'll research areas of drug discovery, which has hundreds plus steps. I am told. We are not healthcare experts, but we do have few healthcare experts in our team, and we are bringing the power of GenAI to see where it can add value or accelerate the process of drug discovery. uh, for example, next pandemic, we should be able to discover vaccines faster and do clinical trials faster. So we still have to research how AI can intervene and accelerate that process. So that will be our goal. We are not healthcare scientists. We are Gen AI scientists coming into the domain of healthcare.
yes, absolutely. Okay, Paul has answered one for the FDA. I think that's what I even aligned with earlier, and some of the FDA regulations are still coming up on that area. So it depends on the context and which they are used, whether they are fall under regulatory definitions. Yeah. Okay. So moving on, understanding the, so this is basically more of like we are going to discuss the process of the drug discovery and what are the sub steps that are associated in the drug discovery process. So if you look at these particular aspect here, so understanding the disease mechanisms, we have genomics analysis, then we have transcriptomics analysis, protein target analysis, pathway analysis, and assistance. This is the understanding of the disease mechanism. That's the first step. And then you have the drug discovery step, which is basically the chemistry in silo simulation, ADMET prediction, and lead optimization. And then you have assistance. And then the fourth step is clinical trial, where you go with clinical practice, patient result, and assistance. So these are the four major steps that is basically involved in the drug discovery process, and especially utilizing LLMs for that drug discovery process. Now, if we talk about some of these like genomics analysis or transcriptometrics analysis, here the idea is to learn about how or what type of LLMs. I'm not going to be like deep dive into exactly what happens in the genomics analysis or what is the previous stages, but yes, I can talk about the technical ability of LLMs to basically understand these genomics process or transcriptomes, right? How do we understand transcriptomes, and how we used to do in the previous version of the language models, right? Like BERT. So there are different types of BERT models that are being trained only specific to genomes or the transcriptomes or proteins, and they are used for doing this kind of analysis. Once we have these base models, right, ready for the process, we can do certain things here. So if you look at these three stages here, like clinical subtyping, then you have target disease linkage, and then you have target validation. So in the first step, which is clinical subtyping, this is the step where you will have this clinical analysis and ethic and regulation compliance. So you would check if the the process has identified a drug that is having a good drugability, right? So it has to get that level of confidence and the compliance score to to get the approval from, right? So the understanding this stage, basically the disease mechanism stage, which is the crucial for the first stage of drug discovery and development, right? The primary aim of these stage is to identify suitable protein target for the potential drug to act upon. So if you look at this, the first step in the clinical subtyping that involves is multinomics data collection. Then you have clinical analysis, and then you have ethics and regulatory compliance. So this starts with genomic analysis, which is having again genetic variants, promoter region prediction, AF site prediction, splice site prediction, and epigenetic mark prediction. Then the next step is basically going with target disease linkage. So if you look at these five parts of the disease mechanism identification, we are going to go with each of them one by one, and then we are going to go with the drug discovery stage, which is chemistry, in silo simulation, admit prediction, and lead optimization. Okay, any questions so far? Okay, so in the process of genomics data analysis, right, the first step, which is basically what do we do in this stage, is basically get the genome wide association. So there is something called GWAS, which is an a multi-decade long study around the genome wide association study, and which is basically known as GWAS in short, that has been identified critical genome regions like various diseases, and that there are significant advancements in that area. So if I show you guys the GWAD, so there are one second, I should write it completely here. Genome wide studies. Okay, so yeah, so genome wide association studies, that's basically if I have hundreds of or basically this study is done on the animals and plants, but mostly and very good base to understand the sequence or and linkage between different genes. Rishabh has a question: are these open source data set to explore? Are there any open source data set? I I would say like some of these models are not really trained with open source, because but the data sets that are available is like more of a specific research organizations they have developed. So they are not really open, unless like if you're talking about the protein LLMs or the gene LLMs, right? So they are not open, Sandeep. Rishabh, there are a number of open data sets which are on Kaggle, which anyone can access, and even open source data set which have a medical data like clinical. Yes. clinical trial and UniProt is there. Any widely used data set for drug discovery specifically? And I think that's what I'm talking about. The drug discovery, you would not see a lot of data set that is available openly as of now. Okay, but if anyone finds it, please feel free to share. Okay, so moving on with the first stage that I was talking about is the genomic analysis. So in the genomics analysis, the first stage is like you find the genetic variant. So you can see here, if I have two different genetic variants, if I can compare between the genetic variants. Okay, so the genomic analysis process is the first stage, as I said, and recently there has been significant interest in this area, like advanced LLMs are being used. Like DNA-BERT is one of the very popular models that has come up, I think in 2021 or 2020, and that paper had discussed about using some of the techniques to identify similarities between DNA and human language. So and that's more of like a structural similarity between DNA and human language. So you can have a specialized training on vast amounts of nucleotide sequences. So if you look at this DNA-BERT, right? DNA-BERT has uh, a BERT specific model, but this has got pre-trained bidirectional encoder representation. But DNA-BERT got updated recently, and that has DNA-BERT 2, which I wanted to have a quick discussion, maybe 2 minutes. Can't spend a lot of time because these, like each of them are like very huge area. So this BERT 2 is an efficient foundational model on benchmark of multi-species genome, and this is having two improvements. One, that it uses BP, bi-pair encoding, because earlier versions had an issue in identifying, or basically the tokenization ability. It was not using BP technique, and the other one is that it has got an evaluation benchmark. So what these people found that the publications and they basically found that there is not a good evaluation model, evaluation criteria or benchmark available on this. So they came up with genome understanding evaluation, GUE, which is a benchmark containing 28 datasets for multi-species genome understanding benchmark. Okay, so this is a DNA-BERT2, which is an foundational model for multi-species genome identification. So if you guys wanted to explore this, there are some data sets. So Hugging Face Model Hub and link to direct download that is available for this model, DNA-BERT2, which is an 11 million parameter model. Let me share the link here in the chat. So that basically says that, like, you can have a task like code promoter detection or transcription factor prediction or promoter detection or splice site detection, which some of them we have seen there. And they basically tried on the species, different species here, you can see. And there is like summarization of genome understanding evaluation benchmark. So that basically gives you the number of data sets, number of classes, and the sequence length that is being used. So this basically also had a huge token length, which was not supported in the DNA-BERT. DNA-BERT has a limitation to use the length, or the sequence length was like too small, which is basically the amount of input sequence that you can provide to the model. Okay, and if you look at this paper, it is kind of gives you an understanding of how BP is giving a better result than the previous encoding or previous tokenization modules or versions. Okay, all right, so moving on. So in the genomic analysis, that's what we are discussing here. DNA BERT or DNA BERT 2 has a good capability in understanding the similarities between DNA and human language. So the first other thing is like, as I said, in the genomic analysis is the genetic variant analysis. So the first you can see in this stage here, genetic variant analysis. What is this? So this is the application of nucleotide LLMs. This actually does an analysis of, let's say, the genetic sequences which follow a specific language pattern. So nucleotide polymorphism, or SNPs, which is basically a single nucleotide polymorphism that has different type of insertions or deletions, or you can have a more complex arrangements, right? And this is basically, again, there are models for each and every activity that you see here, and the terminologies that is being used here, like nucleotide LLMs, right? So there are nucleotide LLMs that are like now coming up humongously, like it has like an LLM based models that are like trying to get into this space where you are using nucleotide LLMs that demonstrates the ability to detect significant functional genetic variants. Okay, and that's basically directly from the DNA sequences. All right. So nucleotide transformer, like I said, DNA BERT. Nucleotide transformer was one of the another specialized nucleotide LLM that also demonstrated the ability to prioritize functionals. So if you now, I'll go to the paper once, because this is what we are actually referring to ultimately, and that gives you the confidence of these analysis, because these are done by these folks, right? So okay, let me do one thing.
Oh, this paper is from September 24.
Yeah, this is a very, very, very recent study. So sometimes this is like these papers are like updated over the time, and they give that idea. But this is submitted on 6th September 2024, and yeah, we are like October 6th today, just a month back. Okay. So and this is basically done, if you see, I mean, just to give you a context, Department of Data Science and AI and Drug Discovery Biology, Monash Institute of Pharmaceutical Science, Monash University, School of Information Communication Technology. So, and Howard, there's a guys are associated with this. So there is a lot of validation that comes in from based on these worldwide journals that are being published, and it is always to have a good reference, because you also have that confidence that the information is being ethical, and what is being published is like it is validated right across. So if it is from a book, I might say that it is solely dependent on the author and what he is writing there, and our trustability for that. So nucleotide transformers, right, which I was talking about, is another specialized nucleotide LLM that has also demonstrated the ability to prioritize functional genetic variants. And it can also be further trained for specific variant identification tasks, such as classifying SARS-CoV-2 variant or understanding SARS-CoV. So basically, these are like, I think if we had during the COVID, people may have used these kind of LLM models for and identifying the genetic variants. Okay. Hyena DNA, one of the another popular LLM framework that is basically push the boundaries of genetic variant analysis by enabling the modeling of extremely long genomic sequences. So you can use up to 1 million tokens to identify the genomic sequences. So Heiner DNA, what it takes is like, let's say, it takes these genomic sequences, and if it is having extremely long genetic sequence, it's able to predict the next sequence, and so on, right? So you guys can look into this Heiner DNMs, like, it's an explosion of information, and I would wanted to keep it limited, while we also touch upon some of the important aspects to this, so that we are able to cover most part of these paper. As you see, like, it's a long one actually, so I wanted to have these like very crisp to understand the major aspects to this. Okay, so Heiner DNA's ability to process such extensive contextual length allows to capture long range interactions in the DNA. This is again on the genomic analysis process yet. Okay, and the other one is genomic regions of interest prediction. Genomic regions of interest prediction is like an it's basically promoter regions, transcription factor binding sites, and splice sites are all some of the crucial elements in regulating gene expression. Promoter regions, transcription factor binding, and splice sites. So this is like having, despite their varied roles, they are all contributing to complex regulations of when or where or how genes are activated or silenced, or silenced, right? So this is the alternate alterations of mutations in these regions may bring about overexpression or underexpression of a gene, potentially causing diseases. So to address this challenge, there are specialized nucleotide LLMs that are being fine-tuned to predict these regions of interest, with the results showing outperformance against previous state-of-the-art method. So there is a Dalla Torre 2023 paper, which again talks about how these nucleotide LLMs are being fine-tuned and is able to predict uh, the, the, the different genomic applications involving the areas of genomic regions of interest prediction. Okay, so there are epigenetic markers that refers to chemical modifications such as DNA methylation or histone modification, and that affects the gene expression without altering underlying DNA sequences. So these marks are playing a crucial role in regulating gene activity, influencing both disease development and therapeutic targeting. All right, so this is the first stage, and then we have transcriptomatics analysis, which is basically more of like, investigating the entirety of RNA transcripts that are organisms or cell system generates using certain conditions. So in this area also there are a lot of developments happening, especially transcriptomics analysis, and the data is often sparse. So the challenge with this is like, there is not much of data, and if the data is available, that's lot of times the data is very sparse, because there are the data is like, having in specific disease states, or like particularly for rare disease and disease affecting clinically inaccessible tissues. So relying solely on these data for specific disease would likely not suffice to development robust and accurate models. So to address this existing limitations, specialized gene LLMs have been proposed. Okay. Okay. The primary technological development in this subfield is specialized transcriptomic LLM or gene format. So last webinar, I talked about little bit about gene format, and which is developed a natively or innovative way to method mapping each single cell transcriptome into a sequence of genes ranked by their expression level. So this is basically known as rank value encoding. I don't know how many of you have seen gene format. So they have trained with 30 million single cell transcriptomes. Okay. that enable context-aware predictions in setting the limited data in network biology. So I've shared the GitHub link in the chat. And there are like SCGPT, which is an again a 2023 discovery, and the other models like SC Mullen and SC Foundation have demonstrated remarkable ability to analyze transcriptomic data effectively as foundational LLMs. Okay, so if you look at these, some of these papers, like as I said, gene former, right? So you would be able to see what are the benchmarks that is associated to that, and how good these models are doing. For example, SCGPT, right? 1 second. I don't know if I can type it here, but that's fine. Yeah. So if you look at SCGPT, so they have basically pre-trained SCGPT model, which is and you can get the download links for that, which is pre-trained on 33 million normal human cells, or for zero shot cell embedding related tasks, or 13.2 million brain cells, or 13.3 million blood and bone marrow cells, right? So you can see how these SCGPT kind of models are being trained, and they have multiple version of that. I think these will keep coming as we see over the time, and this is like a good reference of the models that are more of focused towards cell-based model and specialized model training on a specialized healthcare task. Okay, so it has this flash attention is being used. It's more of like very efficient computation that it provides, especially in the memory and the time that it takes for the attention. As you guys know, in LLM, the most heavy computation that is been done is by the attention process, isn't it? The attention mechanism takes a huge amount of computation. Okay, so we can actually keep talking about different models, but the idea is to get through this process of um, the um, the drug discovery, right? And then we have mRNA expression analysis and gene network analysis. So I'm not going into these in detail. And so the other third stage in that genomic analysis is like protein target analysis. So a protein sequence is often most accessible data about the target that can help explain its potential to play a role in disease mechanisms. With drug discovery, scientists often targeting it as a starting point. So the starting point is like they actually go with the protein target analysis. So if you saw here in this image here, as I said, that we have genomic analysis, then we have transcriptomics analysis, and then we have protein target analysis. Okay, so in the protein target analysis, we have evolutionary conservative analysis, protein folding, functional annotation, protein-protein interaction prediction, and ligand binding site prediction. These are some of the examples of the protein target analysis. Okay, so evolutionary conservations are like, so this is like again using a specialized LLM in protein analysis. So do you remember what we mean by specialized LLMs? Again, you need a domain specific model that is trained on, let's say, nucleotide or genes or proteins, which I in very broadly they are referring as specialized LLMs in protein analysis, as explained in the representative work like ESM. So ESM transformer is one of the famous models again, which is like again available in the Hugging Face also. Sometimes it doesn't allow me to search here. So ESM1B transformer. This is available in Hugging Face. So this is a transformer protein language model trained on protein sequence data without label supervision. That means unsupervised way. The model is pre-trained on UniRef 50 with an unsupervised mass language model. Okay, so this is the UniRef 50 dataset. Like some of you were asking, like if there are, so you can actually go here, and if you wanted to get access to, like I think somebody, Sandeep or someone has mentioned, Rishabh has mentioned about that UniProt, right? So you can see UniProt, UniRef, UniPark. These data sets are that are available, and there are some embeddings also available for these. Okay, sharing the link here, chat. So they are, how they train it, just like normal BERT models. 15% amino acids are masked, and 80% of the cases the masked amino acids are replaced by mask, and 10% of the cases the masked amino acids are replaced by random amino acid. So you know how BERT model works, right? If you have an sequence that is basically 10% of the times are masked, or 15% of the time, you can choose that percentage, but let's say 15% and 80%. So it is able to kind of, we are able to force upon the model to predict the like fill in the blanks kind of thing, right? So in a layman term, and it helps the model to predict the next sequence of tokens and so on. Similarly, here this model is trained on the protein sequences and protein language, right? So, and it basically is able to use this unsupervised way of MLM technique in the BERT methods, and it's a BERT based model primarily. It's not a lot of, like, it's not trained with lots of data. That's one of the things that we need to understand. Okay, so there are some evolutionary conservations, and then if we talk about protein folding, but before that, let me talk about this ESM language model that I said, like ESM 1 billion transformer, which can make accurate predictions of multi mutation effect across variety of proteins with different functions without any additional training. There is other one, that is MSA transformer, which is again a multiple sequence alignment approach, and which is added information from MSA as an input, and MSA transformer enhance the ability to interpret complex relationship within protein sequences and outperform single sequence LLMs. Protein folding is more of targeted using ESM and the MSA transformer. Moving on, functional annotations. So with advancements in general LLMs, protein chat, I'll talk about this protein chat that was proposed to provide an interactive platform where users can upload protein sequences and structures and pose questions about brief description of protein function. Okay, so protein chat is one of the popular, again, models that has came up. Second, sometimes it's because of shared screen doesn't allow me to type here protein. Yeah. So this protein chat or prot chat GPT is implemented chat GPT like functionalities for protein research field. And it works in similar principle with natural language conversation system. Users are allowed to upload protein 1D sequence or 3D structures, FASTA or FDB files, and pose diverse related questions. Okay, anyone have explored protein GPT here? Any healthcare LLM expert? Just checking if anyone have explored it. Paul, so GeneFormer can be treated as specialized LLM. GeneFormer is definitely a specialized LLM. Yes. Paul. It's a foundational transformer, but as it is pre-trained on 30 million, that's a single cell transcriptomes. we can say that it is an specialized LLM, not a generalized LLM. Okay. So like protein chat GPT, right? Or PLP. So this is using a multi-level protein language dataset with sequence, structure, and a description of the dataset, right? So you have a sequence, you have a structure, and you have a description of the dataset. That's what you pass it through 1D sequence encoder, and you also pass an protein structure, that is basically your in input query. That's the first stage, and then this gets the structure embedding. Ultimately, everything that you will convert for the LLMs is to embedding form, and that goes into 3D or 1D, depends on what type of input data that you are providing, and you have a protein prompt, right? Uh, so what is the function of this protein? That's the question. The answer is the protein seems to be a part of insulin, which is a vital hormone produced by pancreas, integral to the body's energy and glucose metabolism. And when insulin binds its receptors on cell surface, so that's a pretty good explanation about what is happening. So even a layman person actually gives a protein structure in a 3D, or let's say in a 1D form. The model is able to get this prompt questions, and the LLM decoder is able to give you the output generated based on the protein description. Okay. Any questions? Anyone? So this is what we are talking about. The LLMs that are like protein chat was proposed to provide an interactive platform where users can upload protein sequence and structures and pose a question. And a recent work in AI for Science and Quantum 2023 also demonstrates the GPT-4 demonstrates considerable expertise in understanding the proteins. Although GPT-4 kind of models are like more of generalized models, they are not specialized model to understand protein sequences, but however, they can also be used for these functional annotation. So what we are saying is, despite these advances, the study of protein language models has remained relatively limited in scope, right? However, ESM2, which is an advanced version of ESM1B transformer, and ESM3 2024, so have been addressing this gap by scaling up to an impressive 15 billion and 98 billion parameters. That's a huge parameter set. I don't know if you have checked ESM3. And ESM3 is like, advanced level transformer based protein sequence model. And I have this issue here. Sorry, I'll try not to have more tabs open. Yeah, ESM3. Okay, so and they have trained with earlier it was like 30 million. Now you can see ESM3, which is an generative model for biology, able to jointly reason across three fundamental biological properties of protein, sequence, structure, and function. So you can see that it takes sequence, structure, SSA, SSA, and function. So, and that goes through the transformer block, and you provide backbone frames, and the transformer block is able to identify the proteins, or I understand the protein. So this is a very much scalable due to transformer backbone, and it also uses has trained with like 98 billion parameters. That's a lot. Okay, small version having 1.4 billion parameters. I'm sharing this link here. Okay, if anyone want to try it out, I mean, I would definitely in our full-fledged course, I wanted to demo some of these models, and we'll have a project that would be more of a description of, that will be more of like an full-fledged project understanding of using these kind of very state-of-the-art models that are available like ESM3 or Protein Chat GPT. Okay. All right. So I shared this link with all of you. And there is protein ligand interaction and binding site prediction. Protein ligand interaction and understanding protein binding sites play a pivotal role in understanding protein functions, right? And interaction, and serves as the essential foundation for rational based drug discovery. So it basically helps in gaining insights into these interactions in crucial and identifying potential safety issues related to target proteins, act as a guidance for the design of therapies. Okay, so protein-based specialized LLMs have demonstrated notable success leveraging their extensive understanding of protein language, and this include prediction of metal ion binding sites or protein-protein binding sites or small molecule binding sites, etc., etc. Right? So protein-based LLMs have advanced with the introduction of AlphaFold multimer. AlphaFold multimer was in paper in 2021. This is basically initial application is to predict the multimeric complex structures, and it turns out AlphaFold multimer is capable of predicting protein-protein interaction directly from protein sequence as accurately as unique protein-protein docking methods that were used. So there is something called doc GPT, which I am going to talk about. Doc GPT is a protein-protein interaction model that was very popular recently, and Doc GPT. You don't have to go to GitHub for this, and yeah. So this is basically a model trained on thousands of uh, Docker image source code. At the end of model is able to generate new source code of Docker image. Now, I wanted to, sorry, so go to protein docking, not the one that is, yeah. So yeah, that's why I don't want to search the GitHub here, but rather want to get the paper. Second, I keep this bookmarks, and it's like little bit of search issue. That's why it's not about allowing me to search this efficiently here. Yeah. So we get to Mac Python and Zoom.
I see a frozen screen. Is it for all, for me only?
Is it showing frozen screen to all of you?
For me, it is.
No, sir. Fine
Okay, it's
from-
my network. Sorry.
Is it working for all of you?
Yes, sir, yes.
Okay, yeah. So yeah, I was searching for the doc GPT, which is more of like, having protein ligand interaction LLM, and more focus towards the protein protein interaction. Let me get that over here. Yeah, now I think I got it. Yeah, I'm going to get a PDF version of that rather than the link here. Yeah. So it's basically site-specific protein docking and design. So this particular model, which is, if you look at these very interactive image here, which actually what it does is it takes the proteins, a different set of protein data set, and this is using an end-to-end deep learning method that is basically having a site specific protein docking capability. So effectively, this is accommodating the whole flexibility of the protein docking ability and utilizing a binding site information compared to AlphaFold multimeter. So this has more a better ability or strength. This basically having an ability to process unbound and predicted structures within the protein docs that are available here. Okay, so I really don't want to go into depth about each of them because each paper is like a lot of discussion here. But if you see the binding interface and interchain context, that basically is the more detail of the protein protein docking protein docking interaction and interface here, and that goes in the invariant input representation. This is basically having this pair chain feature and residue chain features. Okay, and that goes to the main network, and then finally is able to get the complex and confidence prediction for that. All right, so I'm going to share this link as well, which is in having this doc GPT. So DocGPT or MacPerson is an innovative approach to in the protein docking, and this is end-to-end deep learning method stands out for its flexible, insight specific protein docking capability. Okay.
These healthcare GPTs would most likely be narrow GPTs or overfitted to that situation of that paper, right? So they are not certified for generalized use, I'm guessing.
Not all of them, Vivek. So basically, these are like right now in the experimental stage, and
Right,
you would
right,
not see a very like in worldwide comparison and evaluation for them. But
right.
some of the models are coming up like with a good evaluation, like CameCrow and Gene Farmer, right, and Protein GPT, Chat GPT. So those models are like more like in globally accepted and becoming globally accepted. I would not say all of them. So these are like the protein docking is one of the technique that I want to discuss, but these are not like you can use very robustly. And right now, I mean, you can see also like by the things like who is publishing it, so it doesn't go and too popular. Yeah.
Mm-hmm. Mm-hmm.
So just to understand that, okay, maybe so there is a space in future in a couple of maybe years, we would see that this area is evolving more, and we might see more models like AlphaFold Multimeter and or Doc GPT that are coming up with more advanced techniques to interact protein interaction or getting the binding site prediction. So because we are talking about a set of steps here, right? So it's not just disease mechanism understanding is in huge space, right? So we have genomic, we have transcriptomes, we have protein target, we have pathway, and we have assistance. So all of these are like we are just talking about one area of that, and that too we are talking about 2-3 LLMs just to target one sub area of that. So then we have PROT-T5, right? So PROT-T5 is a specialized protein-based mechanical a machine learning model. I mean, it's more of like, I would say mechanical in the terms that it needs to have an, lots of data that you don't really like in case of LLMs, you're able to predict and able to generate things. With ML models, you need to rely heavily on very much structured data and and you need to have labeled data, right? So is but they are able to do predict binding sites and with metal ions or nucleic acids or small molecules and so on. So there is a recent one that is significantly enhanced by Rosetta Fold all atom, and this is in protein ligand interaction modeling with LLMs. Uh, this Rosetta Fold all atom is also another important discovery in terms of how protein interaction can be used or discovered using the LLM. Then we have pathway analysis. In the pathway analysis, GPT-4, because here you can use some of the generalized LLMs, like the capacity of general purpose LLMs such as GPT-4 in analyzing blood transcription null modules related to, let's say, erythroid cells, or demonstrating that these models are efficient in the knowledge-driven pathway analysis. Right. So I think we already have seen like some of the graph-based modules and graph techniques that are being used already to find a relationship between multiple multiple proteins and protein pathways that can be actually, again, sometimes you don't, or maybe the specialized LLMs are not good for that. You can use a generalized LLM to identify the pathways. Okay, so gene regulatory network analysis, that is basically the idea for helping the researchers decipher complex disease pathways. This is one of the other subparts of in this process of drug discovery and analysis. So far I haven't talked about what is the next step, which is like the drug discovery. So let's get into that now. Understanding disease mechanism has to have these specifics. Then let's go to drug discovery. But before I do that, any questions, anyone so far? I know it's a lot, and that's why I had to divide it into two webinars. But it's good. It has really a lot of valuable information that I'm sharing here, and definitely, if you are going to work on these area, these knowledge is going to help a lot. So in the drug discovery process, it starts with hit identification, right? Where the professionals find compounds with potential therapeutic effects. Now, once you have a hit identification, what is the next step? Next step is hit to lead, right? That involves more refined selection from these hits. Okay, so once you have an hit identification, then you go for hit to lead. That involves more refined selection from these hits, identifying those most promising for further development. And then the third step is lead optimization. The third step is lead optimization. Sorry. Okay, so hit identification, hit to lead, lead optimization. So this is the critical process of enhancing a lead compound's efficacy, stability, and safety via editing. So finally, we have preclinical development. Finally, we have the preclinical development. Okay, this is my fourth stage, because I ultimately have to get the final result, right? And okay, so this is the essential or testing, having an essential rigorous testing of optimized lead compound in animal models to assess the suitability for human trials. So it has to get ready for the human trials through the animal, using the animal models. So in this survey, it has begun the outlining the specific downstream task associated with each operation. Following this, we will explore LLMs can be incorporated into these tasks to advance the drug discovery process. So the first step is chemistry. So mechanical, or I mean, in the medicinal, chemistry is essential to drug discovery and development through independent laboratory work and compound synthesis, which is more of like no software involved, more of like a mechanical process, right? You have a medicinal chemistry. And then autonomous lab operations use robotic manipulators, controlled and programmed to execute complex chemistry and synthetic reactions. In addition to high throughput screening requires compounds to precisely and efficiently synthesized as part of the heat identification. So LLMs has proven a highly valuable in this field. What is hit lead? What is mean by lead optimization? Yes, I'll come to that soon, Pramod. I'll come to that. Okay. Especially the lead optimization and what is lead. So heat is basically, you know, if you are trying to get a particular drug to identify if it has an effect, right? So that's where you, if you see an effect, that's where you see that it hasn't hit, right? So this is basically, it finds the compounds. I mean, there are drug discovery, there's already a process that takes a lot of time, as I earlier also said, like let's say 10 to 15 years of time. And in this process, you basically, when you identify a particular disease and you try to see what are the drugs that kind of gonna help in that curation process, right? So it's a long process. So in that, if you are able to find the compounds that are potentially going to help in the therapeutic effects, that's where you find an hit, right? That's where hit. So now once you have the hit, that will lead to refined selection. So let's say if I have 5 or 10 different hits, I would finally go for a refined, or not 10 or 5 but maybe more, and then you get a refined list of the hits from there. Okay. And then finally you do lead optimization, which is basically enhancing the lead compound. So maybe let's say there are some compounds that that gonna help in this, in the disease identification or disease curation process. You might also gonna check if there are other drugs that can be, or other compounds, sorry, that can be used for that identification or helping in the optimizing the lead. Okay. And then you have the preclinical, which is the last stage of it, where you actually are being ready to test the drug that is being identified. So what we are saying is, with some of the chemistry robotics based user requirements, in case of using a code that programmatically do this part of identifying the lead, right? So like Kimcrow is one of the examples of that. LLMs can translate user requirements into complex experimental protocols and convert them into specific understandable robot instructions. So this is like, first stage of using either a programming language, not really in LLM, but LLM can be again used for these lead, to get the hit identification. Okay. And then we have chemistry robotics, which is chemistry is an integral part of conducting chemistry experiments. Like general LLMs like GPT-4 or Code Llama have shown the ability to generate effective code. Therefore, it is logical to use general LLMs to generate robot executable plans, as they have been trained on a vast amount of code. So if you have to generate code that can actually find the molecules and the drugs that are necessary for this hit identification, it can actually randomize to specific code generation for finding a task plan or specific chemistry description language, or even using natural language. So so there are a lot of baselines that are like systems like reader. Synth reader is like an having an accuracy in identifying or like chemistry experiments and reading the chemistry experiments. There are preliminary attempts using GPT-4 to generate compatible scripts in Python to control OT2, a computer controlled liquid handling robot, achieving 95% success with 5 iterations. So so there are many such things that are going on, but I would say these are like in research aspects, so can't give a lot of detail around that. And there are few here and there some experiments, but there is I did not find so far a very solid process that is involved in the process of chemistry robotics. Okay, and then we have retrosynthetic planning and reaction prediction. So this part, and you see that wherever we have more textual information and more structured, in the sense that unstructured information that is available over the domains, or even non-domain, we are able to do better in those areas. So an early attempt of LLMs for retrosynthetic planning is the molecular transformer. Molecular transformer is another, which is like 2019 paper that utilizes simple encoder decoder transformer framework. This is another transformer based model, and the model is trained to take reactants and reagents as input and predict the chemical product that can be synthesized for a reaction. So it takes the reactants and the reagents as input. Okay, so I don't know how many of you have explored molecular transformer here. So there is a search issue for me again. Yeah, I paste that here. So basically, this is calibrated for chemical reaction prediction. So if you, which is basically the heat part, like you were asking, if the model or there is a model that can actually find the chemical reactions, right, that could happen between different molecules, or especially the compounds that are being used in forming a particular drug, it's kind of gives you the first stage of the hit, right? And this is again presents an ML for molecules materials workshop, Neuro IPS 2018. So that kind of gives a little bit of validation for molecular transformer here, and its implement models were based on Open NMT Pi, and the trained model was made available online through a graphical user interface. So if you wanted to explore this IPN hosted model, which is more of like RxN for chemistry, so predict reaction, find retrosynthesis pathways, and derive experimental procedures with RxN for chemistry. So I'm going to share this link, but not going to go ahead with this for now. Okay. So the capabilities of LLMs further extended by K-Informer. K-Informer that I talked about earlier in my previous session also, which is based on BART. And BART architecture is one of the other models that came up around 2020, which is again another way to basically use the encoder decoder, but it has a little bit of changes in terms of how this architecture is being used. So it includes an additional pre-training process that involves reconstructing masked smile or FASTA files, so smile strings, and using an autoencoder to convert original smiles to embedding sounds. Okay, and there are some general purpose LLMs emerged in the field, like such as KMCRO. and Boico. So Boico system uses web search and simple calculations, while CameCrow adopted a more sophisticated approach. CameCrow is developed with I would say a comprehensive range of customized molecule and reaction tools. So that gives CameCrow a better advantage than any other model that is available today. So CameCrow and CameGPT, CameFormer are some of the recent models, and CameCrow is much more recent than the previous ones that I've given examples with. Okay, which is basically helping to find the chemical reactions. Okay, so I would suggest for the chemical reaction, you guys can check this CameCrow paper. It's a very useful tool for the chemistry reactions. And then we have de novo molecule generation. De novo is a process of that involves new molecule structure. I mean, which is basically the task in the silico simulations that involves creating new molecular structures with the potential to be effective drugs, because ultimately that's your goal. Maybe Vivek was asking such models. Yeah, exactly. That's what I shared. So CameCrow kind of models that are being already there. So de novo molecule generation is a complex task in the in silico simulation that involves creating new molecular structure with the potential to be effective drugs, because that's what is finally you wanted to achieve. So this process is categorized into two types: unconstrained molecule generation, which seeks to populate the chemical space of the training set, and constrained molecule generation, where the molecules are synthesized to meet specific desired properties. So constraint generation requires a model to consider various off targets, appropriate psychochemical properties, ADME characteristics. Then we have pharmacodynamics, toxicology, and synthesizability. So to benchmark the performance of de novo molecule generation method, Guacamole was introduced to provide a benchmarking framework considering aspects such as validity, uniqueness, and novelty. Because this is what you need to have good benchmarks. And I think yet the benchmarks are being established. There is not so far, there is not a lot of benchmarks that are being developed in these aspects, because I think this is still an evolving area, right? So it's going to take some time to get good level of benchmarking that is like more of governed by the respective departments and also the government organizations and foundations, because otherwise the trust will not get developed that much. Okay, so Guacamole is one of the benchmarking models for de novo molecular design. De novo molecular design that we are talking about here, and you guys can look into that Guacamole. You wanted to just understand what it means. So let's say if you have a chemical compound and they're generated by Smiles or any other model, and you wanted to basically test out that if there is an activity against the receptors, right? So that basically tells you whether the model, the particular molecule has a hit or no hit, or if there is a lead that can be followed or not, or cannot be followed. So Guacamole was introduced to provide benchmarking framework considering aspects such as validity, uniqueness, and novelty. And specialized language models have demonstrated remarkable proficiency. For instance, it has been have shown the even simple RNN based models can perform exceedingly well on challenging generative modeling tasks. And these models effectively learn the complex distribution of molecules, such as highest scoring penalized log P molecules in Zinc 15, or most significant molecules like in PubChem. So to explore wide and novel chemical spaces, LLMs such as Smiles, LSTM, and Organ have been assessed with the unconstrained generation ability to directly generate an organ, right? So there is a mol GPT, or which is a molecule with GPT architecture, can handle multiple constraints. It is also trained by recovering a molecule with its scaffold properties. Okay. So the reinvent series represents a more advanced approach in this category, and it is sweeping the capable of meeting up to 10 different objectives, including synthesizability, selectivity, etc. So this is the last stage of that we were discussing, and then we have protein ligand interaction prediction. I'm not going into very much detail, and then we have lead optimization. So the stages that we talked about is admit prediction, in silico simulation, and lead optimization. So admit prediction is like the prediction of absorption, distribution, metabolism, and excretion and toxicity, right? So these are the 5 different terms that are being used in the admit attribution compounds, which is critical for hit to lead or lead optimization stages in the drug development process. I wanted to definitely deep dive into these areas and get into more detail of like how ChemBird or PubChem data sets can be used for these kind of admit prediction, and how from hit to lead optimization or lead optimization could happen. And there is the last stage is more of like lead optimization, which is the step to aim modifying the drug candidate molecular structure or protein sequence to enhance its potency, safety, and stability. So once you are able to identify the molecular compounds, the first stage is to hit, right? That's the hit. And then you have the hit to lead, which is more of like you have the essential uh, uh, you know uh, essentials to distinguish the compounds with favorable pharmacokinetic profiles from those with negative characteristics, ensuring the progression of only the most promising drug candidates. Because if you are able to eliminate, and let's say elimination and selection, right? That's what is more of like a hit to lead and lead optimization. And then in the lead optimization, you are going with molecular optimization, more of like protein optimization, and then yeah, so these are the 2 stages that we have, molecular and protein optimization. Okay. And there are some LLMs that are coming up on this space as well, like molecule STM, and chat drug, right? Chat drug and GPT-4, no doubt. So GPT-4 also has some basic abilities to optimize molecular structure, but I think we would need a very specialized elements or LLMs that can help in optimizing this space. Okay. And then we have protein optimization. ESM is one of the, because it's a protein based LLM, and ESM3 having let's say 70 billion parameters can be a good model for the protein optimization technique. And then the last stage is the assistance, which is basically understanding the disease mechanism. And this phase require a diverse array of information sources. Those involved in drug discovery also need access to various relevant resources. Okay, so to gather this information, a procedure known as information retrieval is used. And this is where I think a good model, like, a better model could be these generalized LLMs. And there are some tools like Galactica and GPT-4 that can assist in clarifying scientific concepts and data. And this is where the area that we had, like, which is more of like an assistance, where you wanted to see, like, we have built this CellBot, right, where we are trying to get all the research publications specific to a particular disease. And we also wanted to throw some more detailed analysis towards what are the clinical trials that happened for a specific drug that is being discussed through these different research papers, right? So you need certain tools like Galactica or GPT-4 that can assist in clarifying the scientific concepts and the data around that, right? And this is what is the assistance. Okay, so the last stage of the drug discovery process. Okay, any questions so far? Anyone?
No question, but from my side, I was just wondering when I saw this small GPT, it came out around 2021, as I
Yes.
saw. So you see that from so early, these researchers, different researchers in these fields or other various fields, so they started utilizing these generative models from then on.
Yes.
So early on. I mean, we came to know about these things maybe 2022.
Yeah.
Late, around November or something. But you see, from 2021, even 2020, they started working on all these generative models and started researching on how to leverage these generative models for their research and all activities.
Yeah.
So advanced they are, means getting all the information and utilizing those things.
Yes.
My God.
Yeah. Yeah. So it started actually recently, and you see this this area, like you said, mall GPT, and there are lot of models that are like similar on the similar space, like mall GPT. Then you have Guacamole that I just talked about, and then Moses, right? So these are like some of the very popular. Moses is like a benchmarking platform for the molecular generation models.
Moses, okay.
Yeah. Yeah.
Yeah, go ahead.
Yeah, so basically we need to see more such benchmarks are more important than more models, because over the time you want to see like which models are doing better in which area.
Right.
Yeah.
Right.
Yeah, go ahead, you were saying something.
Sorry, I muted. So yeah, that's what I'm wondering is how they come to know about all these research papers, and it might be helpful for them. I mean, in the research community, obviously, it always propagates all this information very fast.
Yes.
Again, and means I was just wondering, means since we are not from
I
this
think
background.
there are some research tools also available, Paul, that helps them search the relevant research aspects that are happening, and also the current research that is going on, right? So they can actually pull those things.
Yeah. And I see, you think about this small GPT and other things also. So early they started thinking about that one to come out with these things, even before ChatGPT became popular among masses.
Yes.
That's what I was wondering. So 2021 they came out, means they started working on 2020,
Yes,
maybe.
even before. Yes.
Even before, maybe.
Yes. And nowadays papers are like, they are writing papers like in maybe less than 6 months or 3 months sometimes, because a lot of things are like you would not even expect that is there, and it comes up within, and every day I think maybe hundreds of paper coming on only in the LLM space. So
wow.
Yeah, so I think it's very, very competitive for the research community especially.
Yeah, within 6 months if it comes out, that means why? Really?
Yeah,
That's so fast.
they have no time to wait and then publish a good research, because otherwise somebody else will already will publish those kind of things. Like globally, it is a huge competition there. Yeah.
Right.
Yeah.
No, I had no idea about this. Means see all these chemistry, chemistry robots are there for a long time maybe, but the way it has been advanced with using latest technologies and all those things.
Yep.
Oh my God.
So especially this chemical, you said like it is there, but I think especially CameCrow kind of models, right? That came up in predicting different molecules and molecular reactions. So that's a very, I would say, one of the one of the main reasons that these areas are growing, because if you are able to predict molecules and molecular reactions, you can basically build drugs.
Okay, okay.
Yeah, you can build innovative drugs, although that's a still a, like, there are a lot of regulatory things and validations, but you can actually begin and start suggesting. So let's say if somebody is working already in the drug discovery process, and you are able, and they are basically working for, let's say, years, but let's say through these models you are not really finalizing it, but if you are giving them 10 close suggestions or 5 close suggestions, that also saves a lot of time for them. And if they uh, I mean, who knows, they are able to come up with a drug faster than what they were able to do before.
Definitely, definitely. Yeah, at least it helps to aid to think about them. I think about the next step of the research, and
Yes.
yeah, yeah.
Yeah.
That's good.
Okay.
So,
So both ways, reducing the time as well as reducing the cost, it can help you. I mean, even you don't need FDA approvals for that. I would say is, uh, you ultimately, you're gonna need an expert who, or and also, and multiple doctors opinions before they do the clinical trial, because that's the final stage. And in the clinical trial stage, even like when it is discovered through drug discovery process of, let's say, 5 years, 10 years of process, the next stage is also like, in the last session, like webinar, Mr. Nikunj was telling. So we can say that that stage is also a longer process to apply a particular drug on a patient, or maybe start with an animal, or plant based species, or samples, right? So that's going to also be like, we can reduce the time for that using this technique. Like I know that one of the research that is being done with one of the big Indian company in the services industry, and they're having their R&D division, and they're working on synthetic skins, uh, where they actually create these different formulas, right, for let's say skincare, or sunscreen and lotion, etc., etc., and they apply on that synthetic skin, and the reactions are exactly like human skin, and they are able to actually come up with a proper lotion or formula that is more of like, can be given to the humans.
wow, wonderful.
Yeah.
That's good.
So that's already in process, and L'Oreal and big companies are already working with these R&Ds to get solutions like that, so they don't need to directly apply on human skin, but rather they will test it on those synthetic skins.
Awesome, awesome. Really. So many innovations are going on.
Yeah. So clinical trial, I hope over the time we would see maybe artificial patients, so on whom they will apply this discovered drug, and they will come to better safety and maybe better confidence before they go with the final stage of testing with any human sample. Yeah.
Right.
Over the
Even
time.
a digital twin for a human body.
Yeah.
So
Yeah.
it simulates human body and receptiveness to new molecules.
Yes. I think there is a term for that which doctors use for, I forgot the name. The doctors use patients who are like fake patients, but they are humans, but they are basically used for testing a particular drug, or basically or identifying a particular disease, but they are not real patients. Uh, so there is a research paper that I was reading a couple of weeks back, uh, where they are trying to come up with such kind of um, uh, uh, the replacement of those humans, actual humans, to artificial humans.
So they are real humans, you say, but fake patients, something like that.
Yeah, so like, let's say you say like, okay, you go, I mean, you basically be chosen to do that, and you are, let's say, having a particular, you don't have any kind of disease, but the doctor is trying to test out their approaches. It is not a very, like, it's not the research area, but it is something that is already a proven drug, but they wanted to test on a person before they actually give to the actual disease.
Patient.
Yeah, patient. But
Okay, okay.
yeah, yeah,
Riddhi is saying something digital clones. There is another research area, I think.
yeah, digital clone, and I think a digital brain is already happening. I think almost 5 years back I was discussing with one of the persons in California. So he was working with a digital brain cloning technology. uh,
Oh,
yeah, but that time these language models came up, but not large language models. I think now it is much more easier to do that, I mean, in comparison to before 5 years back.
yeah, definitely. Digital brain cloning. My God.
Yeah, so their advertisement, I saw their site. I don't know now, I don't remember the exact site name, but I remember that they were advertising that if you want your grandfather to be alive, you can subscribe to this. So so basically they are taking all the memory, all the things for their grandfather, grandmother, and they're going to have a brain clone for them.
Wow, fascinating.
Yeah, yeah.
Oh my God. Well, at least if I can augment my little poor brain through this way, I will be very happy to subscribe.
Yeah, yeah, I think things will happen soon. Already happening. So
Yeah.
yeah,
Well, that will be a good demand in the market, so you can augment your brain and you can utilize more
yeah.
your brain more effectively.
Yes. Okay, so the last stage is like clinical trial. So which is you having a clinical practice, patient results, and assistance, because this is what you would finally would look for. Phase 3 is the last testing stage performed on the many patients after a optimal treatment selection. So in this phase, the new treatment is compared to existing treatment to understand its differences. And the final phase is post marketing surveillance, during which the drug is monitored for adverse effects. So adverse effect, now also adverse effect detection is like a huge area, and that doesn't require to have like drugs that are identified by these artificial process or LLMs. But even in the real process, we have this adverse effect detection process, right? So I would say like clinical trial itself is a huge area. Even let's say comparing with the manual process, I mean, or combining the manual process plus some part of it using having a drug monitoring or surveillance process. So using LLMs, one of the most significant areas where they can be used is clinical practice, result analysis, and clinical assistance. And the subsequent paragraphs will delineate. So let's go with that. Basically, the first is clinical practice. So coding ICD or matching patients with trials, predicting outcomes, and planning the trial themselves. These are the clinical practices that we are talking about, the responsibilities in the clinical trial. And these responsibilities span various areas and have traditionally been fulfilled by experienced practitioners who rely on their knowledge and expertise. So hence, chemical trial practitioners face the challenge of reading and understanding large amounts of information, such as electronic health records, EHRs, or trial eligibility criteria, ECS. So trial protocols and outcome reports. So fortunately, general LLMs have emerged as a promising solution for accelerating these processes, which is again, I would say we can use general LLMs for this clinical practice to help the researchers find information faster. And then we have ICD coding. So in this The first stage is ICD coding. ICD coding is essential practice in clinical practice, which deals with assigning ICD codes to patient. And these usually a time consuming and laborious process. By analyzing vast amounts of EHR data, electronic health record, LLMs can predict the most suitable codes for electronic health records, and thus enabling clinical practitioners to make more enlightened decisions, streamlining the process. So Sciatal, right, which is a groundbreaking system that utilizes character-aware LSTM-based network to process diagnosis descriptions from hospital admission record more efficiently. Similarly, we have Zai and Zine developed a tree-sequence LSTM network to encode diagnosis descriptions and ICD codes. Some recent LLMs like BERT XML, which is 2020 paper, incorporates BERT pre-training with multi-level attention to encode EHR, and later uses a multi-level classification model to predict ICD codes. There is PLM ICD. There's a BioBERT. BioBERT is there now some time, and a lot of organizations are already using BioBERT for this. And there's a PubMed BERT and Robata PM and so on, right? So for ICD coding. And then we have patient trial matching. This is the second step of the clinical trial when it comes to matching patients with clinical trials. So the process relies on use of electronic health record to identify viable options based on patients' medical history. Historically, this task was performed manually by physicians and data analysts who would sift through patient demographics and pre-screening eligibility factors to pinpoint the most suitable trial. However, this approach can be time-consuming and fraught with errors due to complexity and diversity of the trial criteria. To overcome this, we can use preliminary work, typically include AHR and eligibility criteria into embedding pair, and then calculate the matching score through similarity. So this area also can be targeted using LLMs, and pretty much like you can get a good result here, right? So because we know that LLMs are good in storing information like EHR or any textual information. So an example of these cross-model framework called Deep Enroll 2020 paper, which utilizes BERT to capture eligibility criteria from the text-based patient records and using a hierarchical structure of latent representation. Then we have Compose, which is significantly improves Deep Enroll by utilizing a dual pathway encoding framework. So Deep Enroll, which is here, and in 2020 again they have come up with Compose, which is improving the Deep Enroll model utilizing dual pathway encoding framework and a composite loss function. And this approach effectively separates inclusion exclusion criteria. So you need to have like things that you don't want to include, don't want to, or want to exclude, right? So this is how these kind of models like Compose can be used. And recently there have been several methods harnessing the general purpose LLMs to facilitate patient trial matching based LLMs reasoning ability, like Med Mono T5. It's a 2022 paper and it is using T5 based system. And then we have Instruct GPT, and that actually says that it got achieving about 72% accuracy in screenability, and is not obviously do not have any without issues. So but it says that traditionally it reduces 90% of the workload for these researchers, because these medical researchers spend a lot of time for the clinical trial matching and patient matching basically, right? And then there is a another model like Trial GPT. Trial GPT uses an architecture that predicts criteria level eligibility and provides detailed explanation. And these explanations are aggregated to rank and exclude candidate clinical trials based on pre-text patient notes. And although the trial GPT correlates well with expert annotation, its occasional error highlights the limited medical knowledge. So I think this area is still evolving in a lot of cases, but till that we can use generalized LLMs or general LLMs. Then we have clinical trial planning and prediction. So planning a clinical trial involves several labor-intensive activities, including searching for historical trials, designing trial criteria, and selecting suitable sites. To accelerate this process, general LLMs have been introduced in the domain, right? So here also, like we have trial bird, we have we have guacamole, then we have GPT-4 and all models. So PG entropy, there's a clinical bird, and so on. So it's a huge area. I mean, I'm really not going to detail it out. And patient outcome and prediction is the last stage of the clinical trial process. So if you see the image here, right? So you have these drug candidates, then you have preclinical stage, then you have lead optimization, that hit to lead. So the stage here that comes from proteins, then small molecules and the RNAs, right? So here you would find these small molecules that basically goes through this hit identification process, and then you would finally get the drug candidate. This is your end goal, right? So when you go through this, you can see there is a chemistry in silico simulation, you have admit prediction, and then you have lead optimization, right? In the first stage, where you are actually doing this chemistry part, you have chemistry robotics, or you have retrosynthetic planning, you have reaction prediction, right? This is where you have the chemistry. So once you know are able to do this, so you were using ChemCro or these kind of models to do the reaction prediction. And then you have in silico simulation. In silico simulation, like you can use protein comparison, like I was talking about DocGPT or something like that, those kind of models, are de novo protein generation, or mol GPT, right? So this can be used for the in silico simulation. And then you have admit prediction. In the admit prediction, once you get these molecular properties through chemistry and in silico simulation, you go for admit prediction, where you are talking about these three things, like pharmacokinetics prediction, or toxicity prediction, or psychochemical prediction, right? So you need to know that what are the different types of the diverse effects, or any negative effects or positive effects that can be leading to this molecular property. And once you do that, then you go for lead optimization. Lead optimization is the last stage, where you have molecular so you will optimize this molecule, you will cut, or let's say you add something, and then you do the protein optimization, and finally you come up with the information retrieval and knowledge explanation. So this is where you need these knowledge experts, and we can assist these knowledge experts to with the LLMs to find these matching, or better clinical and clinical trial and patient comparisons. Yeah, so this is the clinical practice process. So where you have the phase 1 is safety and best dosage level in the normal healthy volunteer, and then you explore the effectiveness of and side effects to that, and then you will compare the new treatment to existing treatment, and then you approve the evaluation on the long-term effect, right? And this is where you have ICD coding, you have patient trial matching, you have clinical trial prediction, then you have clinical trial planning, right? And then this is where you get the patient outcome, doctor documents writing, information retrieval, and knowledge explanation. Okay, so I think that's about it, and I'm going to discuss in the next webinar about the LLMs that are like SOTA in the healthcare space, and what are the benchmarks that are available, and which are basically leading in the healthcare space as an LLM, and what are the, because I see there are some questions also around FDA and any open data set that is available. So I'll come up with those information in more depth, and till then I think that's what I have for today. Okay, so we'll come up with this maturity and assessment, improving multimodality, context window, fairness, hallucination. We're going to talk about these aspects also in the healthcare space. All right, so that's about it, and thank you so much everyone for attending this. I'll also plan for more demos over the time. If you want to connect with us, please note down the contact information. Yeah.
Okay, that's great. Thanks. Final questions, audience. Okay, so as you can see, as CellStrat, we are saying let's use our cumulative GenAI expertise and solve healthcare and pharma problems. So that's our endeavor. So in this process, we are developing a platform, CellBot. We are launching webinars and courses. So trying to take our GenAI forte into the healthcare domain. That's our goal. And we welcome collaborators, interns from healthcare and GenAI domains to collaborate with us. So reach out to us. So thank you all for joining. Thanks a lot, Indrajit. Very nice session.
Thank you, everyone. Take care. See you next weekend.
Yeah, thank you. Thank you.